Thank you, Chairman. Good morning. Good morning still, I think, everyone. So I'd like also to thank the organizers for giving me the opportunity to present this subject, which is entitled Dispersion and Deposition of Droplets in a Classroom. It is a work that has been conducted in the frame of a broad program on the diffusion of a virus in indoor environments. So the subject is more or less for indoor environment and indoor air quality in indoor environments. This program has been conducted with a collaboration of partners from France, the CSTB, and from Canada in Laval University. So here. Next slide. The one.
No, the top is the
top is the last one, and the last button is forward.
Sorry about that.
This is the last one.
Yes.
And this.
So as you may know, that there's droplets, which can be droplets expelled from the mouth, can be potential carriers for pathogens such as viruses. So that's why we focus on that subject here, and especially we propose this study because there's a lot of simulation, like computer simulation, for predicting actually the dispersion, the transportation of droplets within indoor environments. You probably know some studies in hospitals since SRAS, since 2003 or 5 in Hong Kong, and in train, but also in airplanes and etc. But there's very few studies on experimental work actually on that, and we really need to have data for this purpose. So our goal, I mean, in the frame of this project, was to study the particle transfer. Particles means droplets, either droplets or residues, aerosols, from an identified source to other occupants. So that's what we call the exposure, and to surfaces. So the final goal would be to recommend in terms of air quality management when the pandemic contamination occur, for example. And so we use a tracer tracking-based experimental approach with a fluorescent tracer. So here, sorry. Yeah, here. You can have a look on this slide on the experimental classroom to study indoor environments. We actually, as I mentioned before, we collaborate with the Center in Research for Buildings, and they have developed in 2008 such an experimental volume, which is about 90 cubic meter large. And so we use this volume to reorganize the furniture into a classroom. So the idea here is to characterize the air inflow and outflow, but also the airflow velocity within the room, and then to experiment at this scale, what we call the real scale, the dispersion of droplets. So our contribution to this project was to implement an experimental device in order to spread out these droplets into these environments. And so we characterized, I mean, in the frame of this project, we characterized the airflow distribution within the room. You can see here on the left part of this slide, the airflow distribution, not airflow distribution, sorry, the air inlets and air outlets within the room. Here are the tables and here are the chairs. And on the right part of the slide, this is different scenarios to relatively to the airflow distribution when contamination occurs. So if you look at this scenario, for example, it means that from the source, the red cross here, almost the entire room would be contaminated with aerosols and droplets. So of course we choose this scenario. It's the most critical one. So after that, yes. Here you can have a look here on the room, and here is the source. So it means that we call the mouse actually. So the mouse is here. So let's say that it's a mouse, but anyway, it gives us the opportunity to inject particles from the double envelope into the room. So to actually manage the preparation of such droplets in a double envelope to be injected into a room. So the idea here is to show you the room with different equipments to measure the airborne concentration within the room or the particle size distribution within the room. But also here on the right part of the slide, you can have a look at the different tables, and on the table there's some black square representing the test surface that we use to collect the deposited mass on surfaces. So at the end, we can have an idea of the concentration of particles within the room and the concentration on surface. Let's say the mass deposition onto the surface. I'm sorry. I think at the end it will be okay. Just to show you a little bit more about the device that we use to generate these particles. These are the particles that I mentioned. You can see that they are yellowish or orange particles because they contain a tracer, a fluorescent tracer that we use as a tracer from the droplets to the dry residues here on the SEM picture. So the idea here was to use an existing generator, which is actually not used for this type of application, and we really changed and readapted this equipment in order to generate or to mimic what goes out from the mouse. So it means in terms of moisture and air velocity, we had to consider breathing, for example, coughing or speaking, which all correspond to different air velocity here, airflow velocity, so 1 meter, 5 meter, 10 meters per second. And then we had to control the size of the droplets. So that was a crucial point actually for that project. So here is the particle size distribution at just after the mouse opening, which was characterized by several authors. Here the particle size distribution is within the size 10 to 60 micrometer, for example. So we choose two different sizes, and with these different sizes to experiment and to see the dispersion of these droplets. Here is a system after adaptation, so it was very different system from the beginning to the end. So also we used actually after sufficient time of particle exposure had elapsed, we could also collect the materials, the fluorescent materials, and using particle tracer analysis, using measuring by fluorescence the signal that we get from the surface or from the air collection samples, and we could also measure the deposited mass on surface. Here is a small calculation to give you an idea of the deposition velocity of particles onto the surface, which crucially depend on the size, of course, of the particles. So I won't go into the details, but this graph shows you the particle diameter as a function of time, and it shows you here that there's quite a stable diameter for residues. And this is crucial also to us because we need to control for sufficient time, like half an hour, 1 hour, 2 hours, the size of the particles. We have to be sure that it's very stable in size. And we also measured at the mouth opening, not the mouth opening, sorry, the droplets size. And we can see here that it's quite monodispersed. So that was our goal. So it was a good news for us. Then we experiment that at a real scale, and at the real scale we also follow the particle or the mean concentration as a function of a diameter equivalent, let's say a diameter, particle diameter, and we follow the dry residue diameter. And then we have a predicting equation to give us an idea of the size of the generated droplets at the mouth opening. So it shows that for us it was a very good result here, because it shows that the residues is quite monodispersed and there's no particle larger than 12 micrometer. Here we compare these results using experiments for different size of droplets. And for example here it gives you the shape of the jet from a mouse, and depending on the size some particles will be expelled from the jet before complete evaporation, some will not. This depends on the airflow velocity. So we use that result, and it gives us, these are simulated results, and for our experimental result it was quite optimistic. Oops. I show here on this slide the particles concentration for different air collection device, what we call the biosamplers. So we collect a certain amount of air, and then we can, into a liquid, we impact this air into liquid, so we can collect the particles, the fluorescent particles, and then measure the air concentration for certain types of particles. That's what we get here. For each trial, actually, what was unexpected at the beginning, but we can see for each trial that the level, the concentration is quite similar, which means that the dispersion of particles is quite homogeneous into this room for each trial. So, and this is also interesting to correlate to the airflow and airspeed velocity that we get in this type of room. So to give you an idea of how the dispersion would be. Oops. As far as the particle mass and surface is concerned, we actually discriminated the particle mass along an axis parallel to the mouth opening or perpendicular to the mass opening. So then we can have an idea of how the dispersion on the tables would be within the whole entire room. So here are the results. So I will just give a short comment, but it's more or less the same. It means that we did not see a really significant difference along an axis. This is the particle deposited mass of tracer as a function of horizontal distance from the mouse. So which means that from 0 to 4 meters, there's no significant difference. So this was interesting for us, and also for vertical surface, which means on the walls, for example, we did not see, we saw a significant lower concentration than for horizontal surface cables. Then the deposition velocity can be calculated. This is actually the mass flux on surface divided by the mean mass concentration in the air. This is the definition of the deposition velocity that we have here on this graph. And the deposition velocity is the same. Actually, it is not that different from one position to another, from one location to another, and for each trial. So here is one trial, for example, the blue dot here is 1 meter per second. It means that the droplets are expelled at 1 meter per second from the mouth opening, and then we have the contribution between the air and the surface. So means that, but they were, all the deposition velocity were higher than the settling velocity that we can calculate with a data sheet, for example, having the size of the particle. So there's, actually I go to the conclusion, because here what we have, the main conclusion here would be to indicate that we found the absence of droplet deposition, even very close to the mouse, at 20 centimeters from the mouse. As I mentioned before, we were expecting to measure the exposure from the mouse to the occupants within the room, so it's an important idea to us. And the exposure seems to be identical for all occupants. And the mass deposition exclusively due to dry residues on surface. So in perspective, I would say that we will experiment with different scenarios. So it means if we change the location of the source within the room, or adding some people, because as you probably know that the people will have an effect on the airflow velocity because of the plume, the natural plume, for example. So, and we will try, not with this tracer, but trials in real classrooms to have to complement actually our understanding for particle deposition and the particle exposure in these types of environments. So I thank you for your attention, and again, thank you to the